Alaikum. Um, so today we'll be talking about cell signaling. Now there are different ways by which cells can communicate with each other. Okay, different modes. There is direct cell-cell signaling, whereby cells can interact with each other via their receptors on the cell surface. If you remember, we talked last time about cell-cell interaction. Selectins as an example, leukocyte selectins as an example of cells interacting with endothelial cells before transversing, before getting into tissues. We also have paracrine signaling, whereby cells secrete a certain factor, ligand, and it binds on a neighboring cell of a different type. Okay. Now you also have autocrine signaling. This is a case whereby the same cell secretes a ligand and it acts on the same cell. Now there's also the endocrine signaling. That entails for a ligand or a molecule to be secreted into the bloodstream, and it travels a long distance before it reaches the target cell. So signaling molecules can be divided into different types. We have peptides and protein signaling molecules. That would include growth factors like epidermal growth factor. So we're talking about large proteins. We're talking about insulin as an example. Or they can be smaller peptides like oxytocin and enkephalins. We already talked about some of these in the biochemistry course. Now, there's also small molecule neurotransmitters. These are mainly derived from amino acids like epinephrine and thyroid hormone. So thyroid hormone is derived from tyrosine, epinephrine as well, and tryptophan is the precursor for serotonin. You will learn about these next year in the neuroscience module, inshallah. Then we have steroids as well. These are small lipophilic molecules that are derived from cholesterol, and examples include estradiol, cortisol, testosterone, and so on. Then we have eicosanoids. Eicosanoids are derivatives of arachidonic acid, like prostaglandins, leukotrienes, and thromboxanes. Finally, we have gases, small molecules with very short half-life, and that would include nitric oxide and carbon monoxide. So these are the lipophilic hormones. Okay, that's how they look like. They are derived from cholesterol. Now, thyroid hormone is derived from testosterone. Retinoic acid is derived from carotene. Vitamin D is derived from cholesterol as well. So now these hormones are lipophilic and they do not require a transporter to get into. So in general, the way they function is that you have this hormone, small hormone, getting near a target cell. It goes through diffusion inside cells. Once inside cells, it binds to a receptor. It binds to the ligand binding domain of the receptor. Now this receptor, when it's not bound by the hormone, is bound to a heat shock protein to preserve it and protect it. Now once the ligand binds to the receptor, it gets released from the heat shock protein and it forms a dimer with another ligand bound receptor. So this dimer then goes into the nucleus, it translocates into the nucleus, and it binds to specific elements on the on DNA. And these are collectively known as hormone response elements. Controlling gene expression. So they can induce expression of certain genes or suppress expression of other genes. That's for lipophilic hormones. So in terms of cell surface receptors, um, they follow the following scheme. So what happens is that you have a receptor on the cell surface, the ligand, growth hormone, cytokine, etc. The ligand binds to the receptor. Something happens to the structure of the receptor, so it changes a little, resulting in induction of a signal to a molecule. And this molecule transmits the signal to another molecule, to a third one, and so on, until you get the final action. Now, the final action can be altered gene expression, or it can be altered metabolism. So you have activation or inactivation of certain enzymes, or changes in the actin in the cytoskeleton in general, inducing, for example, actin contraction. So this scheme right here is called signal transduction. Now, ligands can be hormones or growth factors. Receptors can be receptor tyrosine kinases or G protein coupled receptors. Then we have transducers. Okay, the initial molecule that understands the signal that is coming from the receptor. Then we have effector molecules, and these effector molecules are enzymes. Okay. Then we have second messengers, by the way. These second messengers are produced in large quantities. So we're talking about cyclic AMP, cyclic GMP, calcium ions, and so on. And then we have the target molecule. Again, it can be DNA or a channel or enzyme or whatever. Now, there are different types of responses. Now, you have a primary response. Okay. Basically, what happens, that's when it comes to DNA, action on DNA. So you can have a molecule that is activated, and it acts as a transcription factor. So it binds to certain elements, inducing gene expression, for example, producing other proteins. These proteins can initiate a secondary response whereby they can bind to other elements, regulatory elements on DNA, inducing a second wave, if we can call it this way, a second wave of gene expression, and so on. So you can have a tertiary effect as well. So let's talk about one example of receptor-mediated cell signaling. And here we'll focus on G protein coupled receptors. So G proteins is really a large family of proteins, and they are very important. They are also known as 7 transmembrane domain receptors because they have 7 transmembrane domains. So they go in and out of the plasma membrane 7 times. So this is really a large family, and it is a very important family, considering that about 25% of drugs treating diseases target G protein coupled receptors. So these receptors are bound to a protein known as a G protein. So G protein really is a heterotrimeric protein. So meaning that it is made of 3 subunits that are different from each other. That's why they're called heterotrimeric proteins. The 3 subunits are alpha, gamma, and beta. Okay. Now the alpha subunit is the active subunit. It's the subunit that needs to be activated. What happens is that once you have a hormone or a growth factor or whatever ligand, once you have it bound to the receptor, to the G protein coupled receptor, it interacts with the G protein, inducing the release of the alpha subunit. When the alpha subunit is released, now it gets activated. And the alpha subunit binds to a GTP when activated. Now, then it moves along the plasma membrane and it interacts with another receptor or a channel or whatever, or an enzyme such as adenylyl cyclase. Now, when the alpha subunit interacts with adenylyl cyclase, the enzyme gets activated, resulting in the conversion of ATP to cyclic AMP. Now, it's very important for G proteins to be inactivated, and they get inactivated when GTP is hydrolyzed to GDP. And this hydrolysis is catalyzed by an intrinsic GTPase activity, meaning that this GTPase activity is part of the alpha subunit itself. Intrinsic means that it acts on itself. It does not act on other proteins. So the GTPase activity of the alpha subunit acts on itself, hydrolyzing GTP to GDP. Once alpha is bound to GDP, it associates again with the beta and gamma subunit, waiting for another ligand to bind to a G protein coupled receptor to induce signaling again. So let's talk about second messengers now. One of the most important second messengers is cyclic AMP. So once you have adenylate cyclase activated, it produces large quantities of cyclic AMP. In order to terminate the signal, cyclic AMP is converted to adenosine monophosphate, or AMP, by a phosphodiesterase enzyme. Okay. Now, cyclic AMP can bind to many proteins. One of these proteins is protein kinase A. So it's a kinase, meaning that it phosphorylates many proteins. Now, this protein kinase A is a tetramer. It's composed of 4 subunits. 2 of these subunits are known as the catalytic subunits, and 2 are known as the regulatory subunits. So in the inactive form, protein kinase A forms a tetramer. Once cyclic AMP binds to protein kinase A, specifically to the regulatory subunits, the catalytic subunits get dissociated. Now they're free, they have the catalytic active site, and they can phosphorylate other proteins. So what happens here is that the catalytic subunit can get into the nucleus, and that results in phosphorylation of many proteins, including including CREB. CREB is a CRE-binding protein. Now, because it binds to an element known as a CRE, which stands for cyclic AMP regulatory element. So this protein then, CREB, when phosphorylated, it binds to CRE, and it allows for binding of other co-activators, inducing gene expression. Now, in addition, the catalytic subunit can also phosphorylate other proteins like phosphorylase kinase. Phosphorylase kinase can phosphorylate the enzyme glycogen phosphorylase, and glycogen phosphorylase catalyzes the release of glucose 1-phosphate from glycogen. So you can have action of the catalytic subunit either inside the nucleus or in the cytoplasm. Okay? So the idea here is that if we have, let's say, epinephrine released. Epinephrine is released when there is low glucose level in the blood, or when the body needs energy, production of energy. So for example, it follows the flight or fight response, meaning that if you see a lion, you have to run. Okay, so and that requires energy. So epinephrine is released in large quantities to induce the release of glucose in the form of glucose 1-phosphate so that it can be catalyzed via glycolysis to produce energy. Okay. Now protein kinase A then phosphorylates proteins, activating them or inactivating them. So it depends on the protein. At the same time, these proteins that get phosphorylated must be bephosphorylated, and they are dephosphorylated by phosphatase enzymes. Now, here you have an example right here. You have a molecule ligand that binds to a G protein coupled receptor, activating the alpha subunit of a G protein. Specifically in this case, we have GQ. Okay, so there are different types of G proteins, resulting in activation of phospholipase C. Now, phospholipase C acts on the a molecule known as PI45 bisphosphate, resulting in cleavage of this protein, resulting in two products. One of them is known as diacylglycerol, and the other is known as inositol 1,4,5 triphosphate, or IP3. So you have two molecules in here. Diacylglycerol can bind to a protein known as protein kinase C, and protein kinase C can phosphorylate many proteins. There are many targets. Now IP3, what it does is that it binds to a channel in the endoplasmic reticulum, resulting in the release of calcium ions out of the endoplasmic reticulum. Now there is harmony in the signaling here because the released calcium ions can bind to protein kinase C, activating it. So activation of protein kinase C requires two things, two molecules. One of them is diacylglycerol, which brings protein kinase C to the plasma membrane, and the other is calcium ions, which are needed to interact with protein kinase C to activate it. Now, one of the targets of protein kinase C is actually a protein known as calmodulin. Now, calmodulin, from the name cal for calcium, modulin for modulate. Okay, calmodulin is a protein that requires calcium ions. So when you have the release of calcium ions from the endoplasmic reticulum, now this calmodulin is activated in a way whereby it can bind to kinases, a kinase known as calcium calmodulin dependent protein kinases. And these kinases can then phosphorylate many other proteins, okay, resulting in actin, myosin contraction, for example. These kinases can also regulate the synthesis and release of neurotransmitters. One of the targets is CREB, or the protein that we just talked about, the transcription factor, that is also activated by protein kinase A. This protein can also be activated by protein kinase C. So why are second messengers good? Second messengers like calcium ions, cyclic AMP, cyclic GMP, and so on. There are several reasons. One of them is that these second messengers are free to diffuse to other compartments of the cell. Okay, so you can have like these second messengers being stored in the endoplasmic reticulum, and they can be released out of ER into the cytoplasm. Now the signal can also be amplified. So activation of one adenylyl cyclase enzyme can result in the production of thousands of cyclic AMP molecules. So you have amplification of the signal. The third reason is that these second messengers can be used in activating or, let's say, regulating several signaling pathways, resulting in what is known as a crosstalk between these signaling pathways. So you have, for example, induction of gene expression or expression of genes needed for metabolism to produce energy. And at the same time, you have another activation of another signaling pathway that would induce actin myosin contraction, okay, which also requires energy as well. So you have harmony in activating or regulating different signaling pathways. Now, signaling can also go through tyrosine kinases, enzymes. So it just happens that some receptors have enzymatic activity, specifically kinase activity, more specifically tyrosine kinase activity, meaning that they can phosphorylate tyrosine residues. These are known as RTKs, or abbreviated as RTKs. So you have the extracellular part of the receptor where the ligand binds to, and you have the kinase activity as part of the cytosolic domain or part of the protein. So what happens here is that you have a ligand that binds to a receptor, and the kinase activity is activated. So the receptor can phosphorylate itself and maybe other proteins as well. So this is how it goes. You have a receptor. You have two receptors that are far away from each other. When a ligand comes and it interacts with these receptors, they dimerize, getting the two domains, the kinase domains, close to each other, so they can phosphorylate each other. This is known as autophosphorylation or self-phosphorylation. Once you have the domain phosphorylated, it can interact with other proteins. So why is it that these proteins cannot bind to the receptors? Because the phosphate is needed to induce these proteins to interact. So the domain now can fit the binding site of these downstream signaling molecules. So autophosphorylation can activate signaling by it increases the kinase activity of the receptor, and two, it creates high affinity binding sites for binding other molecules, like I said. Now, some receptors really do not have a kinase domain, but they can associate with a kinase. So what happens is that you have a kinase associated with the receptor itself. When the ligand comes and binds to the receptor changes structure, getting these two kinases close to each other, so they can phosphorylate each other, having the same effect as the receptor tyrosine kinases, the same end result. Examples include a receptor known as JAK, as well as a protein known as SRC. The previous example is true for cytokines. Now, there are other examples of enzymes that can be associated with receptors. So you can have protein tyrosine phosphatases. So these do not phosphorylate, rather they remove phosphate groups. You can have receptors associated with serine threonine kinases, such as the receptor of transforming growth factor beta. And you have receptor guanylyl cyclases. You have protease associated receptors, such as tumor necrosis factor receptor. So we're talking about different types of enzymatic activities that can be associated with different receptors. Now, you do not have to memorize the examples. You just need to know the variety of ways or the various enzymes that can be associated, or enzymatic activities that can be associated with receptors. Now, let's talk about the RAS signaling pathway, because this is one of the most important and most studied and understood signaling pathways. RAS is an important protein since it has been linked to tumorigenesis, cancer, production of various or generation of various types of cancers, such as pancreatic cancer. 95% of pancreatic cancer cases are caused by mutations in the RAS protein. So what is RAS? So RAS is a small GTP-binding protein, meaning that in order for RAS to be active as a protein, it has to be bound to GTP. When it is bound to GDP, it is not active. Now, RAS is not an enzyme, but it does have an intrinsic GTPase activity. So when it is bound to GTP, its intrinsic GTPase activity would hydrolyze GTP to GDP, inactivating it. Now, when RAS is activated, GDP is released, allowing for GTP to bind to RAS, and now it becomes active. Now, the process of exchange and the process of GTP hydrolysis are regulated by other proteins. For example, you have proteins known as GTP exchange factors, or GEFs. What these do is that they induce or stimulate the release of GDP and binding of GTP to RAS. You have other proteins known as GTPase activating proteins. They induce the intrinsic GTPase activity of RAS. We already talked about RAN, the nuclear transport protein, as an example of a small GTP binding protein. There are other proteins that we talked about, including RAB, RO, and RAC. So this is the MAP kinase pathway that is mediated by RAS. You have a growth factor that binds to a receptor. You have receptor dimerization. Now you have the kinase domains activating or phosphorylating each other, creating binding sites for proteins. Now, these proteins can interact with a guanine nucleotide exchange factor, which would interact with RAS, inducing the exchange of GDP to GTP. Now RAS is bound to GTP. Once bound to GTP, it can bind to another protein known as RAF. RAF is a kinase. It phosphorylates another kinase known as MEK. MEK is also a kinase. It phosphorylates another kinase known as ERK. Once ERK is phosphorylated, it can get into the nucleus and it can phosphorylate transcription factors, inducing gene expression. These genes that are induced are those involved in cell proliferation. And this is why, if RAS is continuously activated, if it is mutated and it is continuously activated, it would keep on stimulating this signaling pathway, inducing the production of genes that are involved in cell proliferation, resulting in the formation of cancer. RAS not only is mutated in pancreatic cancer, but it also plays an important role in colon cancer as well. And One of the medications that are now given to patients with colon cancer is a medication that targets RAF, because it was found also in colon cancer that RAF can also be mutated and it can be inhibited by a certain drug. So what ERK does then is that it goes into the nucleus once phosphorylated, and it induces phosphorylation of transcription factors. An example is a protein known as ELK1. Now, ELK1 binds to the serum response element, and it complexes with a serum response factor protein that binds to these elements, specifically SRE, inducing gene expression. And some of these genes can induce secondary response, a secondary response for inducing transcription. Now, in order to facilitate signaling, these proteins that are involved in the RAS or the MAP kinase signaling pathway are all organized. They are bound to a large protein that acts more like a scaffold protein. So this protein can bind to all of these proteins, making activating one by the other easier than each one of them being soluble or diffused in the cytosol. This protein is known as KSR. There's another important pathway that can also be regulated by a receptor tyrosine kinase, and this is the PI3 kinase and ACT or AKT pathway. Now what happens here is once you have the receptor phosphorylated, it can be a binding site for the enzyme PI3 kinase. PI3 kinase is an enzyme that acts on PIP2, a lipid molecule having So another pathway that is activated in a similar manner is the PI3 kinase and AKT pathway. Now, in the same manner as what we've seen with the RAS, you have the receptor phosphorylated, a certain receptor. And now the phosphorylated site acts as a binding site for PI3 kinase. PI3 kinase acts on this lipid molecule, phosphatidylinositol phosphate, okay, which is made of both a lipid component and a sugar component as well, which is inositol. Now, resulting in phosphorylation of PIP2 into a product known as PIP3, so having 3 phosphate groups. PIP3 would act as a binding site, a docking site for signaling pathways, including a protein known as mTOR. mTOR can induce the phosphorylation of a protein known as AKT, which also binds to PIP3. Now, once you have AKT phosphorylated, now it's active and it can act on a number of proteins, phosphorylating them. Eventually, the final effect, the final outcome for inducing AKT is that it induces cell growth and it prevents cell death. Okay, so AKT is also involved in the tumor genesis of cells as well. So AKT can also function to activate the mTOR pathway, and the mTOR pathway stimulates protein synthesis and it inhibits autophagy. So it inhibits cells from dying. So once you have receptors activated, you can have the final outcome of affecting or influencing transcription factors. In fact, the receptor itself can act directly on the transcription factor. An example is what is known as the STAT pathway. In the STAT pathway, you have a cytokine that binds to a receptor, resulting in the activation of a protein known as JAK. And JAK can phosphorylate, is a kinase, it phosphorylates STAT. STAT is a transcription factor. Once phosphorylated, it can form a dimer with another STAT protein, and they both translocate into the nucleus, resulting in gene expression. Now, similarly, you have the TGF-beta Smad pathway that functions in a similar way. Rather, in this case, you have the receptor itself doing the phosphorylation. So TGF-beta as a ligand binds to its receptor, and the receptor phosphorylates the transcription factor. Once phosphorylated, it goes into the nucleus, it binds to DNA, regulating gene expression. Another pathway that is important for you to know is the one that involves tumor necrosis factor and NF-kappa B. So tumor necrosis factor is a ligand that binds to its receptor, TNF receptor, and it induces inflammation and cell death. And this happens via activating NF-kappa B, which is a transcription factor. So NF-kappa B is usually in the cytosol bound to its inhibitor of kappa B. Now, phosphorylation of the inhibitor results in its labeling by ubiquitins and then its degradation. NF-kappa B translocates into the nucleus, inducing transcription of certain genes that induce inflammation and cell death. An important pathway that has been associated with cancer is the Wnt signaling, which is also important in development, embryogenesis, and cell and tissue development. Now, the Wnt pathway, when it is off, you have a protein known as GSK3, which is a kinase. It phosphorylates beta-catenin, leading to its degradation. Remember beta-catenin? We talked about beta-catenin before. It's one of those proteins that bind to ERK adherin, and it links ERK adherin to actin cytoskeleton in adherens junctions. Now, it happens that beta-catenin is also a transcription factor. So when the pathway is off, okay, it binds to a protein known as GSK3 beta, and this leads to its degradation. Now, when the pathway is activated, when the Wnt growth factor binds to its receptor, this results in the release of beta-catenin. Beta-catenin can then translocate into the nucleus, where it can induce gene expression. So, why is it important that you know all of these pathways? Now, these pathways are important in terms of cancer development, tissue development, embryogenesis, and you can understand how these pathways can interact, or how these pathways can induce variety of ways to activate gene expression or induce a certain behavior by cells. But they are quite dynamic. These signaling pathways are quite dynamic. So you do have a feedback loop. Remember, in biochemistry, we talked about feedback regulation, whereby the end product, for example, would bind or inhibit an early enzyme in the pathway. you have something similar in signal transduction as well. And as an example, NF-kappa B, the transcription factor that is activated by the tumor necrosis factor receptor. Now, what happens here is that one of the activated genes, by the way, one of the genes that are activated by NF-kappa B is the inhibitor itself. So when you have NF-kappa B activated, it induces expression of its inhibitor, and that results in inhibiting NF-kappa B to balance the level or intensity of regulation. So there's also signaling homeostasis as a mechanism by which cells can regulate signaling. An example is the signaling pathway of the nerve growth factor. Now what happens here is that this growth factor stimulates the ERK signaling pathway. And this can lead to either cell proliferation or differentiation. So we have two quite different effects. And that depends on the duration of ERK activity. If you have transient activation of the ERK pathway for an hour or less, that would stimulate cell proliferation. But if activation is sustained, then that would induce differentiation into neurons. Now you also have the idea of one growth factor having different effects on cell types. So an example is nerve growth factor. Now the same growth factor binds to two different receptors, inducing different effects. It can bind to TRK-A receptor, stimulating proliferation, differentiation, and survival, eventually leading to neurite outgrowth. On the other hand, the same growth factor can bind to another receptor. In this case, it's P75NTR, inducing either survival or apoptosis depending on the cell type. So it induces survival of breast cancer, or it induces apoptosis of prostate cells. So it's cell dependent really, quite complex. Now there's also crosstalk between different pathways as well. So remember we talked about a growth factor binding to a receptor, inducing dimerization and autophosphorylation. Then you have activation of the MAP kinase pathway, BRS. The same receptor can also interact with PI3 kinase, inducing the AKT pathway, resulting in cell proliferation and survival. Okay. Now there is a crosstalk between these pathways. So you can have, for example, the RAS protein inducing PI3 kinase. You can have the ERK protein inhibiting one of the intermediates in the PI3 kinase AKT pathway. Don't worry about the details, just get the concept. How about integrin signaling? We talked about integrin a lot, and we said that integrins can be found in focal adhesions and they can be found in hemidesmosomes. So what happens here is that you can have, when integrin binds to the matrix proteins, it can induce signaling via a protein known as focal adhesion kinase. And focal adhesion kinase, well, it's a kinase, it phosphorylates many proteins, including proteins that can induce the RAS signaling pathway or stimulate the signaling pathway. So you have the protein SOS. SOS happens to be a guanine nucleotide exchange factor. So it's one of those proteins that would activate RAS by inducing the release of GDP and the binding of GTP. Now, this shows the complexity of signaling and the crosstalk between the different signaling pathways. Now we're talking about G protein coupled receptors and receptor tyrosine kinases. So you can have induction of one would result in the activation of G protein, which would activate phospholipase C. Now, phospholipase C can also be activated by a receptor tyrosine kinase, resulting in branching of the different pathways. And you can see the different crosstalk that happens between all of these pathways. Eventually, you get a harmony in terms of cell response. Now, it's all regulated now, and it's all cell dependent. So it doesn't mean that you have a phospholipase C activated that you will have these different pathways activated in the cell. It all depends on the different intermediates and how they are regulated in these cells. But that gives you an idea for how crosstalk can regulate cell response to effectors from outside. So the thing is, why is it that cells have specific responses? Why do they have different responses to the same growth factor? Well, because they may have different receptors. And I just gave you an example for the neural growth factor. Cells can contain different combinations of regulatory proteins, and these proteins can influence cell behavior. In addition, the final effector can be different as well, okay, which is the transcription factor, for example. A transcription factor may be found in one cell, but not in the other. Remember, as well, in molecular biology, we talked about heterochromatin and euchromatin. So DNA folding, DNA packaging also plays a very important role as well in regulating which genes would be activated and which genes would be suppressed and which genes would not be affected whatsoever.